All right. Well, thank you. I want to first thank the organizers, Anne and Stefano, for inviting me. This has been a really unique conference for me. I want to say that I've been forever transformed, and that the breadth of speaker has been truly amazing. And I think we're going to experience that. So there's a wide range from dry to wet. And I want to say, like, I want a wet side of neuroscience. And I hope this is not going to cause whiplash after what we just witnessed. So in my lab, we're physiologists interested in understanding mechanistically how molecules like dopamine modify the activity of neurons and circuits in the brain to do the very many things they've been proposed to do, and ultimately to modify behavior. And so in biological systems, dopamine is the prototypical molecule that is really thought to lie at the heart of reinforcement learning. So evidence for this came in the 50s from work from Olds and Milner, showing that rats that had an electrode implanted in a specific region of their brain would press the lever more and more and more if pressing that lever led to the electrical stimulation of that brain region. And work over the following decades showed that really what is happening when the animals are pressing the lever is that they're stimulating brain regions that lead to the release of dopamine. And dopamine is the molecule that's, it turns out, is not so much creating a pleasant or warm and fuzzy sensation, but is really acting as a potent behavioral reinforcer, meaning it'll lead the animal to press or to repeat the actions that led to the release of that transmitter more and more. Years later, Peter Dayan and colleagues recognized that the firing patterns or the activity of dopamine neurons in the brain of mammals had key features of TDRL algorithms, namely that these neurons would fire a burst of action potentials to reflect prediction errors, in this case, reward prediction errors that are then used to update future predictions. And fittingly, dopamine itself is a molecule that's known to be a plasticity signal, meaning that it is known to be able to modify the strength of connections between neurons, or also known as the weight of synapses. And dopamine neurons themselves are embedded into neural circuits that, they're embedded in circuits that are known to be relevant to, again, TDRL models, where it is thought to be reinforcing the strength of connections between cortical networks and striatal networks that encode sort of state and value. Okay. Now, at the same time, there's entirely parallel literature implicating dopamine in the control of movement, particularly the invigoration of movement. And this too comes from pioneering work in the 50s from Avery Carlson and others, showing that in this case, rabbits who had been injected with a drug that pharmacologically prevented the release of dopamine in their brain were essentially paralyzed. And then the same rabbits treated with levodopa, which is a dopamine precursor to boost levels of dopamine in the brain, these rabbits could be brought back to life. At around the same time, the astute neurologists had realized that maybe the phenotype of these rabbits were similar to some of the patients they saw in the clinics who had Parkinson's disease, the main symptom of which is a slowing of movement. And so that led them to postulate that perhaps the problem with these patients was a dopaminergic one, which they discovered indeed Parkinson's patients have a degeneration of dopamine neurons. And secondly, that these patients could be treated in part by restoring dopamine level with levodopa. And it is today that it remains the first line of treatment to Parkinson's disease. Okay, but this leads us to a bit of a conundrum, because if you're like me and you like to position yourself in the brain, imagining that you're receiving these dopamine signals, you're then left wondering, how is it that I can disambiguate or distinguish dopamine signals that are meant for learning versus signals that are meant for moving? And for many years, a hypothesis was proposed that sort of really pays attention to the timescales over which dopamine is acting. Namely, is the release and the signaling of dopamine called tonic versus phasic. So if you think of a brain region like the striatum, it might have a certain concentration of neuromodulator, in this case dopamine, that perhaps there's some concentration at rest, and that's brought about by the fact that these dopamine neurons are always firing at a low level, releasing some dopamine, and perhaps at this basal tone and slow gradual changes in dopamine are really important in enabling movement and perhaps changing gradually the vigor with which we move or the modulation with which we act. And then this sort of, relatively flat and interesting baseline is periodically interrupted by these very phasic, sub-second, exciting phasic transients that are presumably driven synaptically by stimuli in the world that signal rewards and things that are worth learning. And these are the prediction error signals that will then allow us to update our models and learn. Unfortunately, a few years ago, as the techniques that we use to monitor the activity of dopamine neurons got better and better, people started to discover that even in a context of simple locomotion, when, for example, in this case, acceleration is plotted in black, as animals go from resting to all of a sudden initiating movement, and the activity of dopamine neurons is measured here in green, you can see what looks like a very phasic increase in dopamine at the time of movement. And that phasic increase in dopamine was really thought to be important for invigorating that very next action. And this has been reported over and over again, including in Drosophila. Here we have locomotor traces of these flies on a treadmill. And if you even look at the activity of dopamine neurons in the fly brain, you can see that there's a lot of activity related to movement in addition to the reward-related signals. And so we're back to this question: how does the brain disambiguate signals for moving versus learning? And another hypothesis that had been proposed at the time is that perhaps dopamine is not the only molecule signaling this. And there's another modulator in the brain that acts as a differentiating factor, and in this case, acetylcholine. Now, why acetylcholine? It turns out that in the mammalian brain, here I'm showing sagittal sections from a mouse, but here you can see where the dopamine axons are and where the dopamine is released in the striatum. And you can also appreciate that this is one of the densest regions for acetylcholine signaling in the mammalian brain. So that's one thing. The other thing to realize, or to appreciate, is that dopamine and acetylcholine can both signal on the same cells in the brain, and that they each have receptors. But interestingly, some of these receptors exert opposite effects on target cells. And so without getting into the details, if this is the pathway that you need to activate to have plasticity and learning, well, you'll notice that the acetylcholine signaling exerts an opposite and blocking effect, if you want. And thus, maybe dopamine serves as a gate for dopamine-mediated plasticity, such that if both modulators increase at the same time, this learning is blocked. But if dopamine increases and acetylcholine is decreased, then you can have learning. And fittingly, many labs have shown over the years, if you, for example, report from the activity of cholinergic neurons, in this case in primates, against at times of prediction errors, clicks or rewards or things like this, they display this characteristic burst pause type of response. But it's really the pause that drew our attention, because it's at this time that we think of dopamine being high, and perhaps here acetylcholine is low. And conversely, in the context of locomotion, acetylcholine was found to maybe be high, and that's also when dopamine would be high. And here, maybe the learning effects of dopamine would be blocked. So a few years ago, a new set of molecular tools became available that would allow us for the first time to directly monitor the levels of acetylcholine and dopamine in the brain using the actual receptors that these molecules signal on. So this is, for example, a human muscarinic receptor or D2 type dopamine receptor, except they were engineered to also contain a fluorescent protein that when it binds to its ligand, it would now fluoresce green or it would fluoresce red. And this would allow us for the first time to actually really look in the brains of these mice to see, is this how the modulators actually behave in the context of, in different contexts. So we can virally express these sensors in the brains of mice. We can implant fiber optics to monitor the activity of these sensors. And in our case, we do this in mice. Our mice are head fixed and are locomoting in a treadmill in an otherwise uninspiring, dark environment. There's nothing much going on. So first, we wanted to make sure that our sensors behaved as we suspected. If we deliver uncued rewards, there's a prediction error and a nice bump in increase in the levels of dopamine in the brain of this mouse. This sort of makes sense. We also imagine the sort of basal tone of dopamine prior to that. These kinds of average responses can be seen across our experimental subjects. This is all good. If we look at acetylcholine at the same time, we see the burst pause type of response, and we notice a nice dip in the level below these baseline levels. So again, This all checks out. It's seen across animals. And if you plot both of these signals together, you can see a nice sort of anti-correlated signal that we wanted to see. That is, at this time, there's a window of opportunity for learning because dopamine is high and acetylcholine is low. And if you have molecular sensors to detect this phasic increase in dopamine and to detect this phasic dip in acetylcholine, you can engage your learning. Unfortunately, when we looked at the sort of single trial responses, which I'm plotting here in gray, you can see that our sensors display significant amounts of noise. And that if, again, I was a target neuron waiting to guess when a reward is delivered simply by looking at either a pause or an increase in fluorescence for this signal, I would have a hard time because it looks like these moments are met multiple times a second on different trials. But there's also a possibility that this is actually noise and that we needed to understand these sensors better. So for that, we ran some control experiments. So now we're just focusing on periods of time when the mice are still on this wheel in the dark. There's really nothing going on. And if you look at dopamine, or at least at the levels of the fluorescence, which we don't know if it's dopamine yet, but these levels are fluctuating wildly. And we did a number of manipulations. I'm showing you one here where we lesion specifically these dopamine cells and we see the signal go away. This tells us that these are not artifactual, linked to movement, breathing, hemodynamic artifacts. They're really linked to they really reflect the phasic release of dopamine in the striatum of these animals. And we can take a power spectrum from data like these and conclude that dopamine levels are fluctuating over a wide range of frequencies, but there's a peak at about once to twice a second. Do we have I would call it a phasic transient. We can look at acetylcholine and see essentially the same thing, wild fluctuations. If we have a genetic model where they can no longer release acetylcholine, we see no signal, so this is acetylcholine. And if we look at the frequency distribution, same thing. We have phasic releases of acetylcholine about twice a second. So now, knowing that there's a lot of things going on outside of what are explicitly cued rewards or prediction errors that we can recognize, we thought maybe what's unique is, again, this very specific timing between both of these modulators. And so we could look at that by cross-correlating both signals together, looking just at the times of reward, and we find this nice, negative correlation that's shifted in time by about 100 milliseconds. And so I'll remind you that that's what the average signals look like. And you can see that these two signals would be maximally anti-correlated if you took the acetylcholine trace and you shifted it to the left by about 100 milliseconds. And so this is what this is telling you. But then the surprise came when we looked at what the relationship of these two modulators was like at other moments during the task, namely when they're locomoting or even when they're just sitting still. And here we find that the anti-correlated nature is maintained as well. Another way to look at it is if we plot the dopamine fluorescence across these different states, if you want, as a function of the phase of one of those cholinergic oscillations. And you see that they're very nicely and consistently phase shifted by about 90 degrees throughout these conditions. And so in summary, what I've shown you here, what we sort of conclude from these experiments first is that the striatal neuromodulatory milieu is highly structured and dynamic. We don't really seem to have this idea that there's a sort of a very basic or boring baseline, and that we have these periodic events that are going to break this monotony and act as very salient signals for learning. And at minimum, this sort of very simple division between tonic and phasic signaling may be too simplistic. But also that these subsequent fluctuations in dopamine, acetylcholine, these phasic signals are not reserved for signaling, again, external rewards. They're also not reserved for signaling actions. In fact, we see them happening all the time. And so we're left concluding that there must be prediction errors for signaling multiple variables, not all of which we know what they are, but perhaps for different patterns of activity, or you might want to call them thoughts, perhaps. And why we think these are still prediction errors is because each of these transients sort of possesses the necessary structure to update synaptic weights. And that's again because these two signals maintain a phase shift relative to one another. And what we know from other studies is that synaptic strengthening or potentiation of synapses, of connections between neurons, requires three things. It requires high dopamine, it requires low acetylcholine, and it requires a striatal neuron to be active. And so if you're a striatal neuron active at this moment in time, then those conditions are met, your synapses will be potentiated. And those conditions are presumably met multiple times a second, and thus you have maybe a much denser representation of rewards, or this may be important perhaps for, sort of shaping a task or in control tasks where you need to have multiple updates as you make your way towards a more complex, to achieve a complex task, you can't wait for the very, very end to get a reward and update. The other thing I want to bring to your attention that I think is very interesting and I would like to investigate in the next few years is that what this also allows you is if you're a striatal neuron that's active now, maybe 100 milliseconds earlier in an environment where acetylcholine is temporarily high and dopamine is low, you flip your plasticity rule and presumably that's a synapse weakening effect. And so we have in the same preparation now a system where you can both learn and unlearn depending on the exact timing of the activity relative to those endogenous oscillations in modulators. Okay, I also don't know how I'm doing on time. Okay, and then the second thing we were that I want to share with you and that we were curious about is sort of questioning the idea that even if dopamine is seen fluctuating at a time of movements, that it can actually play a direct controlling role in terms of invigorating that very action. And we had some questions molecularly. It's a difficult thing for me to explain, but it is one that it's a hypothesis that had gained a lot of traction in recent years. So we wanted to ask, is it really fair? Is it to think that, or can these movement-related dopamine transient control the gain of ongoing motor actions? And so to do this, we also needed, in this case, to design a new behavioral assay because there are many assays have been used in neuroscience, but it's not always clear that it's recruiting the brain areas that you need, that you're interested in. And so in this case, we modeled our behavior on what we know goes wrong in Parkinson's disease because it's in that context that dopamine had been most conclusively implicated in the control of vigor. And so we designed a task where our mice are head fixed, they're holding a lever with their left hand, and they're not cued because Parkinson's patients are actually not, are not most impaired when they're cued, they're most impaired when they have to self-initiate movement. So all we ask our mice is that they hold the lever still for some period of time, and then whenever they want, they can press the lever is loaded, so it's a hard, effortful action. And if it crosses a threshold, then we call it a press. And if it crosses another larger threshold, then we reward them a little bit later for that action. And then we ask them to wait a little bit and the cycle resumes. I'm sorry, this video is not working. And so when you train the mice to do this over a week or so, you'll find that they end up making these ballistic presses every few seconds, some of which are large enough to be rewarded and others not. Okay, so we can characterize these ballistic presses. We can see they're rather fast, they're discrete. We can measure their speed, and we can see a nice distribution of velocities. We can measure their amplitude, and we see that the amplitude that the mice produce the most is the threshold that we set for reward, but we can also see that this is not a particularly demanding threshold as mice are making presses that can be much larger in amplitude. And lastly, we can see that these mice are acting because they're motivated to press for water. I should have mentioned they're thirsty and they're getting these water rewards but if you devalue the rewards by giving them access to water prior to the task, they won't press, they're not interested. They're certainly not acting in a kind of habitual fashion. Okay, so important for us, we wanted to know, well, is this behavior sensitive to dopamine? There are many experiments we did. I'm not showing you here for the sake of time, but here is the performance of our animals before we do anything to them under control conditions. Say during a session, they'll make about 150 rewarded presses. And then we can lesion the dopamine neurons in the hemisphere contralateral to the arm they use to make the presses. And when we do that, they're severely impaired. They no longer get as many rewards, but they still press. If we give them levodopa acutely, like humans, they can restore their performance. And if you wait the next day, when levodopa has washed out of their system, their performance degrades as well. Now, importantly, one of the ways in which their performance degrades is the presses they make are a lot less vigorous, and as measured by a decrease in velocity and a decrease in amplitude. And both of those parameters are restored by levodopa. So again, it shows nice space validity with sort of the presentation of bradykinesia in Parkinson's disease and the sensitivity of this system to levodopa. So having this in mind, we ask now, okay, well, how does dopamine behave in that context? So here we're using, again, photometry to measure dopamine levels using the sensor I showed you earlier. We're imaging dopamine again in the hemisphere contralateral to the limb used to make those presses. And so here I'm showing the average, each mouse, the average of each mouse is in gray and the average of averages in color. For the lever, you see the nice sort of, inward press or the position of the lever here, the threshold is met. Half a second later, they get the reward. And our traces are aligned to when the reward is received. In the dorsal striatum, we see a nice big increase in dopamine, which you would expect. If we omit the delivery of dopamine, we see a dip, as again predicted by these prediction error signals. But I want to draw your attention here to what happens around the lever press. And the first thing to note, which was our surprise, is we didn't really see a nice big increase in dopamine. In fact, across animals and on average, if anything, we see a decrease in the levels of dopamine, which we can quantify and is significant. But I want to draw your attention to the scale of this signal. It's on the order of 1%. And if you remember the traces I showed you before, or the traces I'm showing you here, these are individual sweeps of maybe 20 or so presses that the mice made. Looking at the levels, the fluctuations of dopamine that exist in the brain of that mouse, and you see that they're much, much bigger, many times bigger, right? And so, and in fact, when we relate dopamine levels measured at multiple epochs during this, prior to the press, during the press, right after the press, to the vigor with which that press is made, we find very little to no correlation. If anything, a slight negative correlation, which is only significant because of the number of data points. But otherwise, it explains very little of the variance of the vigor. And I want to say that we've looked in, first of all, that these data are consistent with others in the field, and that if you look in slightly different brain regions, you see slightly different things. But the bottom line is that in almost all cases, although on average you see trends and those are significant, on a moment by moment basis, dopamine does a terrible job of predicting vigor. So that's a correlation. Can we manipulate dopamine levels, go above and beyond, really hit them with a hammer and show still maybe they can have a slight additional gain factor to be added to the vigor of movement? For this, we're using molecules that we can, again, specifically express in dopamine neurons that it can either excite dopamine neurons or inhibit them if we stimulate them, if we deliver light onto them. And so the advantage of using light is that we can be very precise, we can be very fast. In this case, we can inhibit the activity of these dopamine neurons using this molecule while the animals are making the task. We can also calibrate our stimulation parameters by measuring dopamine levels directly. And so we can confirm that on average, when we deliver again light, we can nicely depress dopamine levels, you see, by 10% or so, which is a very drastic dip in dopamine levels in the brains of these mice. And if we then look on a subset of trials where light was either on or not on, at the vigor with which the presses are made, we find no significant difference in either the velocity or the amplitude, suggesting that it doesn't, whether your dopamine is here or here, doesn't really make a difference on the production of that action. We can do the opposite, stimulate dopamine neurons, calibrate our stimulation so that it's at least as big as what you'd experience during a reward, for example. And we also find no difference in that context. Okay. And so with that, and other experiments that I'm happy to discuss later, we conclude a few things. One, that I think the first thing is to set aside the fact that dopamine obeys a fixed or simple relationship with respect to movement amplitude or speed. There really seems to be a variety of signals observed all across the striatum, but that in all cases, dopamine doesn't seem to be either required for making a big, or the sub-second transients are not required for making a fast or vigorous push, and certainly boosting or depressing dopamine transiently is not sufficient for modifying it either. Now, it is the case that some dopamine is required. And I think when you have Parkinson's and you have close to zero dopamine, and it's been shown that you need to have very, very extensive loss of dopamine neurons in order for you to come up with Parkinson's disease, I think we risk thinking, well, if you have no dopamine, you have no movement, then normally more dopamine must mean more movements. But we also have to remind ourselves that in Parkinson's, it's a very different brain. Lots of things have changed, and that you need some dopamine for the brain to function properly. But we certainly cannot conclude that every single whim of the dopamine system is then going to have a very direct effect on the locomotor system. And so related to that, it doesn't really look like dopamine has a very strong or immediate effect on synaptic weights. If it did, again, I think we would see something at the level of velocity. And instead, I think of dopamine still playing a role on the execution of vigorous actions, but perhaps on longer timescales, including mechanisms of gradual learning, updating of synaptic weights. So for example, if you followed multiple pairings of dopamine with a given action, that over time, those synapses would strengthen through mechanisms like reinforcement learning and give rise to movements of either higher or lower vigor. And so with that, I want to thank you for your attention. Again, thank you for the invitation. These are the members of my lab, past and present, first at NYU and more recently at McGill, who carried out some of this work. Thank you.